So I hope all of you utilized your weekend wisely, started studying for exam 1. And remember to use the learning path on the A&P website. There's a study guide on there, some old exams that you can use. Don't just try to figure out what the right answer is, because if you think you're just memorizing a right answer on the exam, when you get on this exam, it'll feel like everything looks the same and you get it all wrong, and that is not fun, right? So what do you do on an exam? What did I tell you guys to do? Go through each and every one. Try to figure out what makes a wrong answer a right answer. Make a right answer a wrong answer. Change the way the question is asked and see how you would look at it. That helps you fill in the gaps of where those holes are. Okay? Then you realize if you didn't get it right, why didn't you get it right? And it's a form of studying, because you should have your notes next to you and flipping through your notes trying to find the right answer, yes? Okay. So I hope you guys tried to get into small study groups if you're not already in tutoring. Yeah, I know tutoring's been a little full. Some of you may drop after the first exam, so if you weren't able to get into tutoring, something may open up afterwards. I'm not wishing somebody ill on that. I'm just saying keep trying to get into the tutoring. And when you get into small study groups, like 3 to 4 people, right? You get too many people, it's social hour, and you don't get the studying done. You don't want that to happen. Let's see. And whether you're in a study group or in tutoring, okay, if you explain something to somebody, they should do what? Say, hold on, let me see if I got that, and you explain it back. Whether it's to the tutor or whether it's to your buddy, but do it to somebody that's currently in the class or a tutor, because they know what to expect. They won't just say, yeah, that sounds good, because then you'll go, yes, I'm ready for my exam. And then you don't feel so good afterwards. So make sure you re-explain it, verbalize it. That will help. Exam 1 is on Thursday, Meyer Health Science, room 106. Starts at 1 p.m. It goes from 1 to 2:15. If you get done early, you don't have to stay and hang out. Bring a number 2 pencil with you and your student ID, because you will fill out a Scantron, which I will provide, but you need a number 2 pencil because it only reads graphite. So make sure it's not a number something other than that pencil or a pen or something else. It's got to be number 2 pencil. Bring your ID card with you, because sometimes you get a little flustered. You want to make sure you have the correct ID number, otherwise it will delay you getting your scores back, okay? Internet students that I am talking to with this disembodied voice, you have a post in your Blackboard account to sign up for the exam. You can start Wednesday at 8 a.m. until Thursday 1 p.m. or take it with the class if you don't have lab at 1 p.m. And in the Blackboard link, it will have a link to sign up at the testing center. And they do not allow walk-ins, so make sure that you cancel an appointment if you don't show up. Otherwise, they will blacklist you, and they won't let you come in anymore. All right, so on the last lecture, we finished up with material that was on exam 1. So today we will be moving on to exam 2. But with exam 2 material, guess what? A lot of it is a review. So this is good. It's good that you guys showed up today. Exam 1 is chapters 1 through part of chapter 4. Chapter 1 was an A&P overview, learning new terms and definitions. You guys are starting to learn a new language, right? The language of healthcare. Levels of organization, homeostasis, review, positive and negative feedback. This is like me reading the study guide to you, right? I hope this is sounding familiar. Body planes and sections, cavities, linings, quadrants, regions, right? Okay. Chapters 2 and 3 were beginning chemistry. We talked about potential and kinetic energy. And hopefully you guys are applying these terms to what a chemical bond is doing. Like if you're building a bond or you have a covalent bond, it's a potential energy. But if the bond breaks, it's doing what? It's become kinetic energy, and it is releasing energy or giving it off, okay? And we talked about various bonds. We did ionic bonds, covalent bonds. What kind of covalent bonds do we have? Polar and nonpolar covalent bonds. You should know what that means. If you don't, go back and study it, because you really need to know that for exam 1. And it's important for the rest of the material of the semester and in 228 as well. We also talked about hydrogen bonds, which were quite important and also helped you with special properties of water, acid and base mixtures. We got into biochemistry. We talked about our main big macromolecules, if you want to talk about them. What were they? Carbohydrates, lipids, proteins, nucleic acids. Okay? Hopefully you guys know examples of each of those. And we also talked about the molecule ATP. What's ATP stand for? Adenosine triphosphate. Take a look at that molecule again because we do talk about it a lot. Then we started chapter 4, biology of the cell, and we touched on the basic parts of the cell, which we get into a little more depth today. Membrane structure and junctions. We talked about all kinds of different diffusion, right? Passive diffusion, facilitated diffusion, a special kind of diffusion called osmosis, which is what? Crossing through a semi-permeable membrane. Water. And we talked about tonicity a little bit. And then we talked about active diffusion. And there were two kinds of active diffusion. There was primary and secondary. Primary means you need what? You need ATP. It's talking about, like, an example would be the sodium potassium pump. Exactly. Good, good, good. And secondary active diffusion, did it require ATP? No, it did need energy, but in a different way, using the concentration gradient. And so what I did was, in secondary active transport, we had two examples, which is where I pulled the notes back up from before. We had symport and antiport, and I gave you guys a homework assignment. Do you remember? No, you don't? Oh, no. All right, so I said, remember, we were running out of time at the end of lecture, and I said, look at this picture here. And I said, let's take a look at it. See, we talked about the definition, we verbalized it, but I wanted you guys to try to draw it out. And I said, when we came back to class today, we would pull this up. This is on exam 1, so I want to make sure you guys understand this. So to the right, I have drawn in two cell examples. The line in the middle here of both of them is a semi-permeable membrane. So what did that mean? Some things can go through. We're selective. And when we're talking about osmosis, we usually mean only water can pass through. And based on the concentration gradient of the solute, depended on where water would flow, correct? But it's also going down its own concentration gradient. And when I say it goes down a concentration gradient, what do I mean? It's going from high concentration to low concentration, because it's trying to spread out where it has more space, right? We talked about the dance floor, letting somebody in or out. That was a great analogy. Okay, so I drew a circle in the middle. That is an example of our protein. So one of these examples, we'll put number 1 and number 2. One of these is a symporter protein, and one of these is an antiporter protein. Okay, so based on what we talked about before on symport and antiport, and I even pulled it up so you can see it. Okay. And let me move it up just a little bit. And we said secondary active transport was move substances. One of them is going against its concentration gradient by harnessing the kinetic energy, that is not ATP in this case, from moving another substance down its concentration gradient. So two substances are crossing the membrane. Okay? One is going down its concentration gradient, meaning what? High concentration to low concentration, till it reaches like an equilibrium, yes? And the other side, it says, is going against its concentration gradient. So which way is that one moving? It's moving from low to where it's already high. That's why it needs energy. Because why do we need to shove it where it's already high, right? Okay, so this is, now, the difference between symport, sim, think same, and anti, against. The difference is sim means both substances are moving the same direction. Anti would mean one's going in one direction, the other substance is going in the opposite direction. So based on the two pictures, example 1 and example 2, okay, so we have substance X and substance little o, right? Some of them look like O's, some look like funky circles. Okay, so if in example 1, if we move it to, if we move the circle in example 1 from an area of, what did I do here? In example 1, if I move the circle from high concentration through the protein to an area of low concentration, and I move my X substance from an area of what on this side? Side A is low concentration. I want to move it to this side where it's already high concentration. Okay, so the X is going against its concentration gradient. O is going down its concentration gradient. They're going in the same direction, so what kind of protein is this? This one's a symporter. Okay, if we look at example 2, and I say that X is going to go against its concentration gradient, but I'm going to move O down its concentration gradient. They're going in same or opposite direction? Opposite, so it automatically makes it now an antiporter protein. And they did a prettier picture of it over here, of course. But I wanted you guys to try to draw it yourself. Why? Because on the exam, if you draw a picture, it will help you immensely on an exam. You can draw all over that sucker. You can cross off bad words that are wrong, not bad, but wrong words. You can circle things that are important, things that you want to remember. You're answering the right question, not what you think the question is asking you, but what is the question actually asking you. You can draw all over that. You can draw diagrams for yourself. It will help. Okay, so I highly suggest you read a question, you see something like antiporter or symporter, draw it out before you read the rest of the question, then answer it. That way you don't convince yourself to answer what you thought it was asking you, okay? So let's make sure we do that. Does that help you guys? Does that help you guys understand anti and symporter better? Okay. So what else do I want to tell you? Now, where we left off, so then we talked about vesicular and bulk transport, exo and endocytosis. Those require ATP. That was a review. Okay. And then some examples of endocytosis, bringing it back into the cell. We had phagocytosis, which was cell eating, and we had pinocytosis, which was cell drinking. Okay, so cell eating, that would be an example of like a macrophage or a phagocyte that is finding some bacterium or something out there that it brings into its cell so it can digest and break it down. Okay, Pinot, I said like Pinot Grigio, drinking wine, cell drinking. So that's easy to remember. I know it seems silly to remember these silly extra things, but it actually locks it into your memory better. You guys might laugh now, but you'll remember this years later going, silly Dr. Z said pinocytosis. Okay, we talked about resting membrane potential, then we started going into cytoplasm. So from here on down is exam 2 material, but you're going to see that some of what we talk about today actually will help review and lock in some of your exam 1 material. So now we're in exam 2 stuff. We talked about cytoplasm, cell-forming material, organelles, membranous and non-membranous. We have endoplasmic reticulum, which we said was a sac-like network of membranes with one continuous lumen. So both the rough endoplasmic reticulum and smooth endoplasmic reticulum are actually connected with their lumen. It's just, what's the difference between smooth and rough? Which one makes proteins? Rough, because rough means it has what on it? Ribosomes. And anytime you hear ribosomes, you think protein synthesis. I'm making proteins, okay? So that's a review. Smooth endoplasmic reticulum does not have ribosomes on it, thus it has a smooth texture to it if you want. And for the most part, you just need to know that it makes lipids. It does some other things with detoxification, but we're not going to go quite into that detail right now. But no ribosomes, makes lipids. Where do we need lipids? Membranes, our phospholipid bilayers, yes. It's making these for us, which is fantastic. We need that. So, what else do I want to tell you? Golgi apparatus, we left off with that. I think that was the last thing we talked about. Yes? No? Maybe? I think so. Because we started just a little bit of exam 2, I thought. No? We did not talk about this. All right. New material, Golgi apparatus. This is also going to be near the nucleus. So you have the rough and smooth endoplasmic reticulum around your nucleus. Okay? And this makes sense, right? Because what did we talk about with our nucleic acids? What are our nucleic acids? DNA and RNA. DNA stays where? In the nucleus. RNA is made where? In the nucleus, and then it goes where? Cytoplasm, and it finds what? A ribosome. And we're going to get into the details that we didn't cover before for exam 1. In exam 2, we're covering the details of this process. So we had DNA in the nucleus, which was like our what? Our Joy of Cooking cookbook, right? It has every single recipe your body needs to make everything. But we don't want to lug this recipe book everywhere we go, right? We want to make a protein. So we will read off one little message, and we called that what? We want one recipe card, not the whole book, our messenger RNA. So we read that off the DNA, we make our little messenger RNA, our little index card for one single recipe, and that then will go from the nucleus out the nuclear pores into the cytoplasm, where it's going to find a what? A ribosome. And the ribosome is either going to be free floating just out in the cytoplasm and it will make proteins, or it's going to be in that rough endoplasmic reticulum where it means it's going to make proteins, right? Yes. So after we make this protein, and we talked about for exam 1, what does our protein structures look like? We had primary structure, which was just the amino acid sequence. So in a moment, we're going to talk about how we make that amino acid sequence, how we put that, link them together. And what's in between the amino acids? What kind of special bond did we call it? It's a covalent bond, but more specifically because it's a protein, it's called a peptide bond. So that peptide bond, we're going to talk about how that's made in a moment. And as that protein, it only has like the primary structure when it leaves the rough endoplasmic reticulum or a ribosome. And then look here for Golgi apparatus. So this tan color here is part of the rough endoplasmic reticulum. And it has, it's kind of hard to see maybe in this picture, but can you guys see it has little bluish looking gray things inside of the RER lumen? Okay, and then look, number 1 shows that it makes a vesicle. What did I tell you the membrane of a vesicle is made out of? Phospholipids. And then look, this vesicle travels, and the Golgi is actually really close. So the Golgi looks like pita bread that's right up next to the rough endoplasmic reticulum. So this green pita bread is the Golgi apparatus. The vesicle that is carrying a primary structure protein is going to merge, or it shows number 3 here, merge with the Golgi apparatus. And remember, even with endo and exocytosis, I talked about how the membrane merges and lets its contents spill out, or it pulls it inside of the cell. Okay, so the membrane of that vesicle from the RER merged with the Golgi, and it spills its contents into the Golgi. And I told you guys for exam 1, the Golgi apparatus is kind of like the UPS system, right? We drop off a package, we're going to let them repackage it for us, and then we're going to send it off either out to the cell, so it's got to go to our outer membrane and spills out there, or it'll do something else within the cell. So it's showing you a couple arrows like it could travel somewhere else into the cell and do another job. So what I'm going to tell you now for exam 2, in the Golgi apparatus, when we're repackaging things, what did we say happens after primary structure of a protein? It then goes into secondary structure, which is what conformations? It's 2. Alpha helix or beta pleated sheets. And then from that we can go into a tertiary structure, which is what? Secondary folding up on itself. So you may have both alpha and beta pleated sheets kind of in one protein, one globular protein, one polypeptide chain. Both secondary and tertiary structures are due to what kind of bonding? Hydrogen bonding. Easy to break, easy to make, right? And then from there, we could either stay as a tertiary protein or we could become a quaternary protein. Okay? All of this folding and remodeling is happening in the Golgi. This is our repackaging. We might even attach a couple of carbohydrates and some other things onto our protein that we can take off again as well. But this is that repackaging process. So Golgi apparatus is a flattened stack or pita of sac-like membranes. So this is the processing plant for material from the RER, and most of the RER is giving us what? Proteins. Now you know that story. Pretty cool. You already knew all of that. You just had to put it together in a story. So it says for the Golgi, it processes proteins from the RER, and we add or remove portions of the protein before we export it. So we might take out a couple of those amino acids that we didn't want in there. We're going to kind of mature our protein, if you will, and make it better. So an example might be that we might have something called proinsulin. Pro meaning before. So this is the protein that we have made for insulin, but it's not refined yet. We haven't made it the one that will actually be insulin. And it'll go through the Golgi and we will process it. We will remove some things, take off the pro part, if you will, and make it the mature insulin protein that can then be released and sent out of this cell so we could do exocytosis so it can get out into our bloodstream. And did we say that insulin is like the key that opens the door of the cell to allow food in? If you don't have insulin, you can't get glucose into the cell. So this is what we're talking about. So proinsulin would be processed through the Golgi, and when it comes out, it will be the mature protein insulin ready to go. Okay? So we can also form the secretory vesicles for protein export, and those are the green. So that green vesicle is part of the phospholipid membrane of the Golgi. Remember, these phospholipids are not covalently bound to each other. They're just attracted to each other by their hydrophilic heads and hydrophobic tails, right? Look at all the stuff we're reviewing for exam 1. This is awesome. You guys know this stuff. You're going to be ready. All right. So, and then that merges and becomes your outer membrane of your cell then, right? And then it might become an inner vesicle when you take something in like phagocytosis or pinocytosis or endocytosis, right? The main generic term. So lysosomes, membranous sacs of digestive enzymes. So they're showing you a lysosome right here. So again, it has a phospholipid layer, but it is containing something inside of it, something special, like digestive enzymes. Not quite the same ones that you have in your stomach, so think on a smaller scale. These are digestive enzymes to digest. So when this lysosome merges with another vesicle that's come into your cell, it could be from your Golgi, it could be something that was endocytosed and brought in. It merges with that vesicle, spills its lysosomal, what do you call it? I just told you. Digestive enzymes into that other vesicle and starts digesting and breaking it down. It breaks bonds. It can recycle things. So we see here it says we can have a phagolysosome. So those are, lysosome combines with a phagocytic vesicle. So remember, phagocyte pulled it inside the cell, endocytosed it, to destroy, say, a bacterium. So you have this bad guy, this bacterium, out there trying to attack your body, and you bring it in, and then you can merge with a lysosome vesicle inside of your cell. The lysosome spills its digestive enzymes into that merged vesicle and digests and eats up that bacterium so that you can't eat it anymore. And in 228, you will learn that we can take those digested parts and stick them on the outside of our cell, and we let our immune system know, kill this. This is the bad guy. Here's a piece of him. Look, hunt, and find and get it. But that's 228. But it's super cool when we get into that. Super awesome. All right, so we could also do autophagy. What does auto mean? Self. Phagy, eat it. Eat yourself, right? So this is lysosome would digest damaged organelles. So give me an example of an organelle we've already talked about. Any organelle. Like a ribosome or a mitochondria. Right? Let's go with a mitochondria. Let's say mitochondria has been, what did we say he was? The powerhouse of the cell, right? Magic school bus? Yes? Okay. So we have our mighty mitochondria. And he's been working so mighty hard, and he's getting a little old and tired. And so he's starting to break apart. And so we could have a lysosome merge with this part, bring it into itself, and let its digestive enzymes break down the parts of the mitochondria that it chewed up, and then it can recycle those parts. Because what is the mitochondria made out of? Did we mention? It's a double membrane. We're going to talk about them in a second. A double membrane organelle. Phospholipids. We could use them again, right? Why waste what we already have inside of us? Takes less energy to recycle it. So that's autophagy. And then we can also have autolysis. So sometimes, so auto again means self. Lysis means to cut, right? To cleave it. Okay, so self-cutting. So not self-cutting, don't do that. Autolysis, so damaged cells will die if a lysosome ruptures. And we end up calling this a suicide packet. So you have this lysosome, and an example of this would be our nerve cells when they have lack of oxygen. Okay, so nerve cells, lack of oxygen, or O2. So for example, let's say somebody has a heart attack, okay? And yes, we want to try to get their heart started again, but what we're more interested in is how long does it take us to bring this person back to life, okay? So the heart stopped. We're not circulating the red blood cells because the heart's not moving right now. Oxygen isn't getting transported around to the other various tissues throughout the body. If we go more than, say, maybe 10 minutes or so, okay, then our nerve cells have lack of oxygen. And then all of a sudden, the lysosomes inside of there don't like it. And they start rupturing. And when a lysosome ruptures, it's not merged with another vesicle. It's spilling its contents into the inside of the cell. Do you think it's selective on what it's going to destroy? No. So it's been released inside of the cell. It starts chewing things up and digesting things, breaking things down, breaking bonds. That is not good. So if you bring the person back soon enough before these lysosomes start rupturing, the person may come back and be fine. Maybe a little rehab going on, maybe a little fatigued and tired, but they'll be okay. But sometimes we bring people back, and if there was too long of lack of oxygen, they're not quite there. Okay? They might have where they can't recall things very well. They may not have full function of their muscle cells because the nerves can't tell it what to do because they died. Okay? So our nerve cells are particularly susceptible to lack of oxygen, and it will go through something called autolysis, and that's why it's called suicide packets, because it's literally digesting itself and killing itself. And it's not going to say, oh, wait, never mind, I've been brought back to life, I'm going to stop. No, it's been spilled out. It's just destroying everything at that point. So that's why it's sensitive to that. All right, and let's see, where are we now? Look, it's the mighty mitochondria, the powerhouse of the cell. It provides most of the ATP. So you will have many mitochondria per cell, okay? And remember, for exam 1 material, we said that some cells will have much more mitochondria than others. So for example, heart muscle has to work 24/7. It can't stop contracting. It's got to work. It's got to have a lot of mitochondria. Needs a lot of energy all the time. Versus, say, your skin cells, right? They're hanging out. They're literally just waiting to die, sloughing off, things like that. But they don't need as much mitochondria. They have mitochondria, but they're not going to have as many per cell as a myocardial cell, a heart cell, right, would have. And it says it can divide as it contains its own DNA. What's really cool is this DNA. Did you guys know who gives you your mitochondrial DNA? You read it in the news anywhere?
Mom.
Your mom! Yes. So you could totally know who somebody's mom is, not just by checking their other DNA. But this is, your mitochondrial DNA only comes from the mom. Doesn't matter if you're male or female. This is from your mom. Okay? So it's definitely a matrilineal line. And it says it can divide as it contains its own DNA. So we'll talk about that more in the future. But it's a double membrane structure. That membrane is made out of, take a wild guess, phospholipids, yes. And it also contains enzymes for ATP production. Enzymes are what? Proteins. So it has proteins on its membrane that help you make the ATP. And remember, we said this is where we get the majority of our ATP from. And when we go into detail about this, not right now, but soon, a couple chapters away, we're going to learn the exact details of those proteins and how we get ATP from that process, okay? So for now, you just have to know mitochondria is the powerhouse of the cell, contains enzymes that makes tons of ATP, and double membrane structure, kind of looks like a kidney bean, okay? And we will talk more when we learn about the how we make that ATP, how we generate it. We'll talk about the different parts of the membrane and areas of the mitochondria. So we're going to learn the mitochondria in depth in a couple chapters. Oh, there's our little ribosome guy. protein factory, we said, right? And I said you could have it free floating in the cytoplasm or attached to an ER, which would make it a rough endoplasmic reticulum. And both, doesn't matter if it's free floating or RER, both make proteins. That's its job. You hear ribosome, protein synthesis. So the ribosome is actually two subunits. We have a large subunit and we have a small subunit that will combine and make your ribosome that you guys understand as a ribosome. So you have to have both the large and small come together in order to do the protein synthesis. You can't have one and not the other. They both have to be there. And we call it the workbench for protein synthesis because if you had a workbench, you have like your tools on there and you have like the things that you're working on and making or taking apart. So that's why we call it a workbench. This is where we make our proteins. You're going to learn that, remember I already mentioned that we have mRNA and I said that was called what? Messenger RNA. You're going to learn that there's another one called rRNA. What do you think the R stands for? Ribosomal RNA. So these ribosomal subunits are made out of RNA. It's ribosomal RNA. A small, shorter chunk makes the small subunit, and a larger chunk of our RNA makes the larger subunit. And then they come together, and we're going to read our messenger RNA like a ticker tape. And it's going to feed through this area in between the large and the small, and it'll go in this direction. And then when we finish reading that mRNA ticker tape, our message, our little recipe card for one single recipe, what did I say that recipe is going to make? Yeah. A protein. Yes? A protein. So it reads that messenger RNA, that one little recipe to make our protein. And as the mRNA comes out the other side, we are going to use another thing that you're going to learn about in a second called tRNA. Look at all these RNAs we're learning about. You only have to know these three. tRNA is called transfer RNA, and you'll see it in just a moment. The tRNA does just what it sounds like, transfers. It's going to transfer an amino acid and link them together. And when we link amino acids together, what did we make? The protein. We have a primary protein structure, right? Amino acid linked by peptide bonds. It's all coming together. How cool is this? Okay, so we're going to talk about rRNA and tRNA in a minute. I'm just introducing them to you right now. So if we look at cytoskeleton, we talked about that guy for exam 1 a little bit. Cytoskeleton, structural proteins for support and movement inside of the cell. And movement is kind of a key word here, okay? So for movement, what two molecule components of structural proteins did we talk about before, like in exam 1 stuff? Elastin and collagen, exactly. Okay, so if you have elastin and collagen, this though, we're going to talk about 3 parts of cytoskeleton. It could be a microtubule, an intermediate filament, and a microfilament. Okay, so use the names to help you understand a little bit what they're doing. Microtubule is a hollow protein tube. It's involved in the cell shape and also moving things around inside of a cell. Okay, so this is what provides, remember I said there's like a highway system inside of the cell? This is what tells the vesicles how to move like from the Golgi to the outside of the cell. Or when something's being endocytosed into the cell, they're going to follow something like a microtubule saying, follow this pathway, follow this highway or roadway, and I'm going to take you where you need to go. I'm going to usher you in, okay? So this is the highway system in the cell. This is the roadways. And this is found wherever movement is involved. That's why I said movement's the key word here. Intermediate filament, intermediate means like in between, right? So that's the in-between size. So it's in between the microtubule size and the microfilament size, so that's kind of cool. It's a tough protein. It's part of the support system, and when I say it's tough, it's because it's keratin, or it can make up keratin, which helps make skin cells tough. We need these guys to be tough, right? We don't want them to just fall apart at the little brushing up against of something, right? We'd look like Swiss cheese all the time. And then we have microfilament, which is a thin, so think micro like micrometer or, you know, teeny tiny. So think thin strand, very tiny strand of actin. Anybody know what actin does? Have you heard of this before? It's a protein. Helps with contraction. You're going to hear it with, yes, with muscle contraction. It's going to be with myosin and actin. And when we get into muscle physiology, again, a couple chapters down the road here, when we get into muscle physiology, you're going to learn how myosin and actin work together to contract or shorten your muscle, or decontract and relax, not decontract, but relax, and allow your muscle to elongate again, okay? So again, microtubules, intermediate filaments, and microfilaments are under the category of cytoskeleton. So think about what skeleton is. The structure, right? So that helps you remember. Cytoskeleton, cyto, it's in the cytoplasm, so we know we're within the cell. Skeleton, these are the structural things that help give your cell support and shape and help with movement. If we look at centrioles, they are made of microtubules. So what did microtubules do? Moving, right? So this is telling you that centrioles have something to do with movement if they have microtubules in it. So these are found near the nucleus, and it ends up being part of cell division. We're not probably going to get to cell division today, but in the next lecture. So Thursday is your exam. When we come back on Tuesday, we'll probably be ready to start with cell division. And that's where you're going to learn more about the function of the centrioles. But it's found near the nucleus because what's inside the nucleus? DNA, right? Deoxyribonucleic acid. This is our blueprint for the cell. And what do you guys know about DNA? Just not having learned a whole lot in here yet, but what do you know on your own about DNA, besides it being the blueprint of the cell? You guys ever heard of chromosomes? That's what your DNA is when it's wound really tight. And it tightens up like that. We're going to talk about the details of that in a second as well. It tightens up really tight so we can move it around. We're going to move it around with centrioles because it has to do with movement. So that's why it's part of cell division. We have to line up our DNA so we can divide our cell and have the right number of chromosomes going in the right direction. That's what we're talking about there. So this is when a cell is going to divide. We're going to be looking at centrioles. And let's see, what else? Did you guys also, in lab, you should have already looked at the whitefish blastula. Okay, so what were you looking at with that slide? You were looking at mitosis. What's mitosis? Cell division. This is what we're talking about. You can't see the centrioles under the microscope, but you can see what the centrioles were doing. And again, we won't get to talk about mitosis until the next lecture, but we're going to cover that in more detail. So microtubules are part of the cytoskeleton to move things, part of the centrioles to move chromosomes, and they're also part of cellular extensions formed of, dun, 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 there's microtubules again, which means what? Movement again. And we have two things we can talk about. Cilia, not silly, but cilia, which are hair-like extensions that move things. Oh, look, there's that word move again. And flagella, only found in the human in the sperm cell. There are other organisms that have cells that have flagella, but the only cells that we have, or not me because I'm a female, but that males have in the human species is a flagella is what? The sperm cell. And it's a whip-like shape. So cilia only moves things in one direction. Cilia is going to move things across the cell's surface, okay? Flagella is moving an actual cell to get it from one location to another. And if you guys remember sperm cells, you can understand it's got to travel, right? Okay, so cilia only goes in one direction, and in reality what it does is, the cilia is this hair-like extension on the outside of your cell. It will go back, cock itself, and push things along. And then it relaxes and recocks, and pushes things along, relaxes, and recocks. And what kind of things do you think it might push along its cell surface? Could be mucus, like in the trachea. So we had trachea as an example here. So for example, if like me, when I had gotten sick and really lost my voice, and still haven't gotten it back, so I probably was producing plenty of mucus down my trachea. Where's your trachea? It's your windpipe, right? And where is it leading to? The lungs. Do I want mucus clogging up my lungs? No, that would be a really bad idea. So what direction do you think my cilia and the trachea need to move this mucus? Up and out, not down. We don't want it to go down because that would take it more into the lung area. We want it to get out. So it's going to cock and relax and push that mucus up, and cock and relax and push more mucus up. And then the nice thing is, because your body's pretty amazing, it's going to come up, and you're going to even see in 228 that as it gets up into the upper part of the trachea, it's going to have an area where it can have a connection to your esophagus. Your esophagus is where your food and liquid goes, down into your stomach. You can think of it as a sewer system, right? Because you put stuff in and then it's going to come out, literally go to the sewer system. You guys need to laugh more, okay? So this sewer system, because mucus, we don't want to keep it, so it's not very polite to cough and hack it and spit it out into the audience, right? Because that would get you guys sick with whatever I had. I don't want that to happen. That's gross. It's against the law in some places to spit, by the way, out like on the ground, like Arizona. Tuberculosis, anybody? How not to pass it along? Okay. So I don't have tuberculosis. I don't want you guys to think that. All right. So if I can get my cilia to get it up there, I can then swallow it down the esophagus. And then guess what? Your stomach has enzymes and high acidity, and that's going to break that stuff down. So it's not going to damage or get something else sick in the rest of me. And then it's going to go through my intestinal system and end up coming out the other end into the literal sewer system. So that's why we call that kind of like your sewer system. So cilia, move in one direction, moving things along. What might be another example of where cilia is pushing something along?
Question, does your esophagus have cilia pushing down or no?
No, it's smooth muscle that we'll end up talking about when we get into the muscle section. So not cilia in that area.
Isn't there cilia to the eggs?
Yes. So I put the example oviduct, but you could also say fallopian tube. You might be more familiar with that term, fallopian tube. So the fallopian tube has cilia in that ductwork, if you will, so it can help bring that egg that was just released, okay, so ovulation, and bring the egg down towards where? Where do we want to send this egg? No, it came from the ovary. Good guess, though. To our uterus, right? Now, somewhere in the fallopian tube, we're hoping that that sperm cell that has a whip-like projection on it called a flagella is going to get that sperm cell up to the fallopian tube to find that egg and fertilize it. And you'll learn at the, more in 228, that the fertilization actually occurs in the fallopian tube, not in the uterus. And then that egg still has to get pushed down by the cilia to move it down towards the uterus, where hopefully it can embed if it's fertilized and then start to grow and become a fetus, okay, and multiply. And so when we learn about meiosis, which is more 228, it's a little different than mitosis, which is what you saw with the whitefish blastula. Yes?
So when you look at the fallopian tube pregnancy, is that because you don't have cilia in the egg?
So what she's asking is if somebody had, say, an ectopic pregnancy, meaning that the egg implants in the fallopian tube instead of traveling down and getting to the uterus. You cannot develop your baby in that area. When you learn in 228, what's going to happen is that egg is a parasite, and it will search and find a blood supply to feed it, which if it made it to the uterus would have plenty of blood supply there where it's supposed to implant. And it's okay that it's a parasite and implants there because we want it to. But in the fallopian tube, if it ends up having these enzymes that burrow through the layers of the cells that are through there and trying to find the ovarian artery, which is just on the other side of the fallopian tube. And it will start to grow, and then it would end up being too large. It doesn't have the capacity to stretch in the fallopian tube to house and nourish a growing, developing fetus. And so typically what they do is they would have to remove, that's not going to end up being a viable child. It's not going to be able to grow to term. But the problem may be, and there could be very many different reasons why it could happen, but one potential reason might be that the cilia wasn't functioning properly and didn't move the egg along. So yes, that could happen. There could be a whole host of other reasons why, but yes, that could be one potential reason why the egg didn't get to the uterus like it was supposed to. Good question. All right, so that was talking about the cell. Now we're going to get into details of the nucleus. And guess what? Here's our old friend DNA and RNA that we get to talk about again. A good review from exam 1 material. So nucleus, we said, contains the DNA or blueprint of the cell, right? So this is our Joy of Cooking cookbook. Inside of our nucleus, the DNA has every single recipe you need. All cells have a nucleus except one. I rewrote it on here, and that's the red blood cell. So red blood cell did have a nucleus at one point before it matured, and then it kicked it out. So red blood cells do not have a nucleus, okay? Which makes it better for transporting oxygen anyway. It needs room for that. And as you guys know, nuclei take up a lot of space inside of a cell, yes? Yeah, because it's like the biggest thing you can see under the microscope. So here is an electron micrograph picture. Remember I said they look kind of grainy because they've thrown electrons at it to make that picture and bounce it off of there. Here is our nucleus that we're talking about. We're going to talk about some of the parts within. And I'm going to leave these pictures up as I talk about some of the parts within that nuclear structure. So we have nuclear envelope is a double membrane. It's actually 2 lipid bilayers. So how many layers of lipids do we actually have? If a phospholipid bilayer is 2 layers of phospholipids, what's 2 phospholipid bilayers? 4, okay? So you actually have 4 layers. Why do we need 4 layers? This is so important. We don't want things getting to our DNA. We don't want our DNA getting out by accident. We have to protect the DNA at all costs, otherwise we have problems. Okay, these are the instructions for everything. We don't want this going willy-nilly or crazy, okay? So we need to protect it. That's the extra layers of protection of double 2 lipid bilayers that are fused at points creating nuclear pores. And what did I tell you nuclear pores are for exam 1? RNA. So RNA is made in the nucleus like the, I'm spitting on people now, sorry. RNA is made in the nucleus, but it doesn't stay there. It's got to go out, and the way it gets out is through these nuclear pores. So nuclear envelope controls movement of material between the nucleus and the cytoplasm, right? We decide if something can go out or in. We're small enough that we don't let large things in. We're uber selective. We really don't want hardly anything coming into our nucleus, right? We do have some proteins that can be in there, but we don't want things coming and going like our regular cell membrane, our plasma membrane. So we have something called nucleoplasm. What does that sound like? Sounds like cytoplasm, but instead of being in the cyto area, which means just the cell, we're saying it's the nucleo area. So nucleoplasm is like the cytoplasm within the nucleus, so fluid within the nucleus. And then we have something called nucleoli. That's the plural. So a nucleolus would be a single one. So we have a nucleolus. This is like the blueberry in the center of your larger blueberry, the nucleus. So a nucleolus is inside of a nucleus. And nucleoli is the plural of nucleolus. So it has 2 L's instead of nucleus. It's 1 L. So nucleoli is the site of where our RNA, which we said R stood for ribosomal RNA. So this is where we make the ribosomal RNA. These are going to be for what? The large and small ribosomal subunits, okay? And we also have DNA. So we have ribonucleic acid, we have deoxyribonucleic acid, and it says the DNA is 46 separate DNA molecules with associated histone proteins. So the histone protein is like these little orangish salmon-colored dots here. And histone proteins are there whether the DNA is coiled super tight or whether it's relaxed. But it's those histone proteins that allow that coiling. So think of it like a spool, and think of the DNA like the thread. So we can wind our DNA around this spool super tight for the chromosome shape, which is B, or we can be uncoiled and relaxed, which is chromatin, which is A. So both of those are just describing what shape the DNA is in, okay? If it's in the chromatin shape, it means that the DNA is being used. So when we briefly talked about Joy of Cooking Cookbook, we read off one simple recipe. We had to unzip a little piece of the DNA to read off the one recipe off of it. That has to be in the chromatin form because we're using the DNA. As opposed to when we get ready to do cell division, we need to tightly coil it up so we can move it around with the centrioles, which are made of microtubules for movement, right? So the chromatins are tightly coiled around the histone protein, okay? And so they're trying to show you that they have chromatin, which was relaxed. It's winding tighter and tighter around these histone proteins, which winds even tighter into a really, really super tight shape until you have the chromosome shape at the top there. Now, do you guys remember if we lined up all of our DNA together, how much I said it would be inside your cell? 6 feet. So when we wind, if somebody could close that door and stick an eraser in it so people can get in and out, that would be wonderful. So if we wind up the DNA super, super tight, or even when we're relaxed but kind of bunched up, but we're just not super tight, that's 6 feet of nucleic acids stuck together and wound up in a specific size and shape, bless you, that we can fit inside of the nucleus of our cell. 6 feet inside of that little tiny nucleus that you need a microscope to see. Pretty cool. Just saying. All right. So chromatin versus chromosomes. Chromatin uncoiled, chromosomes tightly coiled so that you can move your DNA, because you'll learn that we've got to split the DNA apart. Now I said we had 46 separate pieces of DNA. Okay? Which means, do you guys know who you get your DNA from? From mom and dad. Half from mom, half from dad. So if half is from mom, how many chromosomes is coming from mom? So we have 1 through 23 chromosomes, individual pieces from mom. And we'll have a 1 through 23 set of chromosomes from dad. If you add them together, you get 46. Okay? And so what we say is we have 23 pairs of chromosomes because they pair up based on their size. So 1 and 1 pairs up, 2 and 2 pair up, 23 and 23 pair up. Okay? Or you could say we have 46 individual pieces of DNA. Okay? And they decided to number them based on their length. So number 1 was the longest piece of DNA, and the 2 number 1s lined up, and they said, we're going to call those DNA number 1s. Okay, so that's how they decided. We're not very creative as anatomists, right? So, alrighty, so what else do I want to tell you? So we have chromosomal pairs. We named them 1 through 23, 1 set from mom, 1 set from dad, and we're going to talk about in more detail in just a second, or more with cell division. What do we do with those various pairs of chromosomes? Okay. So where do we find histones? What are histones? It's a protein that the DNA winds around. Okay. So histones are a protein. The DNA winds around it. It's what helps it coil more tightly or not. But it's there even when it's in the chromatin form, the relaxed form of DNA. So let's look at the function of nucleus and ribosomes. Okay. So this next part of the story is actually called gene expression. Where do we find genes? Genes are just little segments on that DNA that we just talked about. So when I say that we're going to read off a recipe card, I'm talking about we read off one gene, one segment of the DNA that tells you how to make one particular protein. And everything is very specific, right? Okay. So when I say gene expression, for example, if we want to look at the protein that might be involved with the shape of our hair, okay, whether you're curly or whether it's straight, or maybe you're an in-between and you're a wavy, right? Or if I want to see about the protein that decides what color pigment my eyes are going to have. Okay? This is reading off one single recipe card off of that. Expression means what you physically see. So you look at me, you see blue eyes, you see, not my real hair color because I color it, but you would have seen my real hair color for what's actually being expressed. What my proteins are saying, this is what it's going to look like. That's expression. So gene expression is like this big umbrella picture of, I'm going to take that recipe from the Joy of Cooking cookbook and I'm going to get it to the point where I make my protein and let the protein do its job. That whole thing is called gene expression. There are 2 parts to gene expression. There is transcription and translation. Okay, both of those make up gene expression. So the term transcription sounds like what to you? Transcribe, or to write down, or you could think like a transcriptionist, like court documents, they're typing out everything in shorthand what the people are saying. So transcription is like copying the message. So what do you think I'm doing in transcription from what I described earlier? Copying the DNA, meaning I'm going from DNA to RNA. I'm making that messenger RNA. Okay? That is what transcription is all about. Translation, what does that sound like? Think of like if I were speaking Spanish to you and you needed somebody to translate what I said into English. So translation is a language change. What I'm doing is I'm talking about the language of DNA nucleotides, and I'm translating it into an amino acid language, which is what? Protein, right? Amino acids linked together give you primary structure of protein. So we're going from a nucleotide, which is RNA, a nucleotide, that's the monomer for RNA, and I'm translating it into a whole nother language called amino acid language. So think translation, language change. So translation is all about going from messenger RNA to making the protein. And we've learned a little bit about the parts, the players, the actors that will do this. Who's going to translate that messenger RNA into a protein sequence? rRNA, ribosomal RNA. The ribosome translates it. tRNA, which we talked about barely, we're going to see it in a second, tRNA is going to transfer those amino acids onto linking that amino acid chain, making the protein. So we have this umbrella of what we're going to do. Now we're going to learn the details of transcription and translation. So if you get lost in the terminology or what's happening, go back to the big picture. Transcription, then translation. DNA to RNA transcription. We're just transcribing the message. Those are both nucleotides, so it's the same language still, right? You're just copying down the info. And then you're going to go from RNA to a new language called amino acid. That's the translation part. Super easy. So go back and remember the big picture if you forget this. But now let's look at the details of actual transcription. So we have synthesis of messenger RNA. Okay, so I wrote messenger RNA, just writing out what the M stood for. Initiation sounds like the beginning, right? So this is the segment of the gene. Remember I said the one little part on the DNA that we think is important. This is our little recipe card, right? Joya Cooking Cookbook, we don't need all those DNA recipes. We just need the one gene off of one piece of DNA. So it's a segment of gene on the DNA is exposed. So this is where I said before, we unzip a little piece of our double strand of DNA. We're going to read off the template strand. Template means like the original. Who's the original? DNA is always the original. So the template strand, when you hear that terminology, you should think DNA. So we have our DNA strand. We are making a copy, one little message, one little segment, messenger RNA. So initiation means we open up the gene on DNA. The enzyme that does it, ENZ, I shortened for enzyme. The enzyme that does it is called helicase. Look, ACE. ACE tells you it's what? It's an enzyme. It's also a protein, right? Yes. Okay. So helicase's job is to unwind that. Remember, DNA is a double strand and it's kind of twisted up, right? So helicase's job is to attach to the, it's going to have an area that tells it attach here to read this gene. This is the one you're looking for. Attach here. It unwinds that part of the DNA and it unzips. What do I mean by unzip the DNA? It's breaking the hydrogen bonds. Those are the weak bonds in between the ladder rungs, right? Between the one base and the complementary base on the other side. We're unzipping that. And it has to keep it apart or it will do what? What do hydrogen bonds do? They form and break all the time, right? So if I don't keep it apart, it's going to try to do what? Zip right back up. But we don't want that yet. We got to read off our message first. So RNA polymerase, look, another enzyme, ACE. RNA tells you it's going to make RNA. Attaches to the DNA promoter area. Promoter just means this is the beginning of the gene. We're promoting it. We're going to start it, right? Of the template, template meant what? DNA. That's the original strand. So RNA polymerase attaches to the DNA, the start region, we're saying, the DNA promoter area of the DNA strand, okay? And then we need to do something called elongation. Elongation sounds like we are doing what? Making something, right? Making something longer. So elongate means RNA polymerase, there's that enzyme, the same one we just said a second ago, builds messenger RNA molecule. It does this by adding RNA nucleotides to the growing messenger RNA chain, and it says in a complementary fashion. Remember when I said RNA is double or single-stranded? Single-stranded. So for a short time, it's going to complementary base pair to DNA just as it's being made, and then that message will come off and be a single strand and do its job, go on, find a ribosome, correct? So I wrote a short segment of what here? Because it has a T, it tells you it's DNA. Okay? So what would be its complementary strand? What would be, so if this is DNA, we have now our messenger RNA being made, and it's going to be what pairs with T? A, U, because it's RNA, it has uracils, no thymines. Then it has G, C, C, U. All right? So you know that that upper strand is DNA because it has thymines in it. T's are thymine. You know that the lower one is the complementary RNA strand because it has U's in it, uracils. You guys remember the other nucleotides, I hope, right? A is adenine, C is cytosine, G is guanine. You are so ready for exam 1. So ready. Okay. So for a short time, because it's complementary base paired to this, what is holding this to the DNA strand? Hydrogen bond. So a little bit of hydrogen bonding going on until we break it again, right? Nice and easy. What are these bonds right here? Covalent bonds. Difficult to break. They're strong. They don't just break. Okay. So let's see. Now we have our DNA template strand. We've now made our RNA strand, okay? And once the transcription, right, this is the transcription, we copied the DNA to a complementary RNA. They're still nucleotides, that's why it's not a new language, it's just transcribing the message. Okay, we're going to do this process until termination, where the RNA strand would be released, meaning we don't have to keep those hydrogen bonds, that'll come off, and this whole strand can now leave. It's not going to stay hanging out with DNA. It's going to be made and leave. Now, if we still need to make more of these strands, right? If we say we need lots of protein for insulin, keep making protein, we will keep the DNA strand unwound and keep reading off messenger RNA segments, pieces that we're making over and over again, okay? But once we say, oh, blood glucose has gone down, we've gotten all the glucose into the cells, we no longer need insulin anymore, you may stop production. Then we can do what to DNA? Let it zip back up. Okay, so it will rehydrogen bond with its complementary other side, coil back up, it's happy, it does its thing. Okay? So, termination says RNA strand is released. That means RNA polymerase, the one that was making our RNA nucleotides, these are RNA nucleotides, the individuals, reaches the end of the gene. It says, oh, this is the end of the message. We don't need any more. This recipe is done. And it will release the messenger RNA and cause closing of the DNA gene or segment and reallow the helix shape. So it twists back up, okay? Which is awesome. That's exactly what we want it to do. So we have this messenger RNA, and it's still hanging out in the nucleus. It hasn't left through the nuclear pore yet into the cytoplasm. It's in the nucleus, and we need to go to the cytoplasm. But what did I say hangs out in the cytoplasm before? Last lecture. Nucleases. ACE means enzyme. It's breaking down nucleotides, nucleases. So if we just let this messenger RNA float on out to the cytoplasm, it's going to get destroyed. So we have to mature it a little bit. We have to make it the right messenger RNA to get out so it'll be a little bit protected until it can find that ribosome so it can make what it's been designed to make, to get that protein made. So we don't want it chewed up. So what we need to do is we're going to do what's called modification. We are going to splice it. So there's going to be parts on this message that we made. Maybe we decide we didn't need so many nuts. So we said instead of 3/4 cup, we're going to go with a 1/2 cup of nuts. And maybe we don't need, some of these nucleotides here maybe don't code for something. So we're going to splice them out and remove it and allow G to connect to U. So we're going to modify this guy, mature him a little bit. We take out some of these pieces, and then we have to add what's called a cap and a tail. So one end is going to get a cap, and one end is going to get a tail, okay? And part of what we're doing is I put the word faux nucleotides. You guys know what faux means? Like pretend, fake. So we're putting extra nucleotides on this cap and tail so that if a nuclease finds it and starts chewing up at one of those ends, it's okay. It didn't get to the message yet. It didn't get to the actual recipe. We're okay. So that's why we did this. So that's why it says faux nucleotides with the cap and the tail. It's preparing and maturing the messenger RNA for the cytoplasm to find the ribosome, to give it time. Okay. So I'm going to scroll down a little bit here so you can see the picture. So they're showing you in the picture here, we have our DNA molecule. We have our helicase, which they didn't really show on there, but helicase does what? It unwinds the DNA, and then they've kind of shown it where it unwound it here, but it's already moved on this direction, and it is breaking the hydrogen bonds that were in between those guys, right? We broke them, so they're not there anymore. Okay. And we're helping keep it apart until who comes into the story? No, another enzyme. RNA polymerase. Okay? Poly, many, RNA, putting many nucleotides on, right? So we're keeping it unzipped, and there's like a little green ribbon that they're showing here on just one side of the DNA. You could be reading either side of the DNA, and it could be happening at the same time on both sides. But we're reading off this one side for this purpose, for this picture. And so we're making this RNA strand right there, okay? And then when we are all done, we've reached the end of the message or the gene, RNA gets released, because RNA polymerase hit the end or the termination part of it. RNA gets released. DNA can recoil back to its helical shape. The message, we're going to do what to it now? We're going to modify it. So we are going to splice out some of the parts. We're going to re-ligate it back together, glue it back together so it's one piece again, and we're going to put a cap and a tail on it. And now it's mature and it's ready to go out through the what? Through the nuclear pore into the cytoplasm to find a ribosome. All right. So I'm just going to briefly talk about translation. When we come back after exam 1, next Tuesday, we will come back to this and do it more in detail. But I just want to give you the end of the story real quick before we finish. So translation, we said, was the synthesizing of the protein. That portion of gene expression occurs in the cytoplasm. So again, you're going to have an initiation area. So this is where you're going to say, this is where you start reading the message. Okay? It's going to, let's see if we have a picture down here. Here, we have a picture. Okay. So it's going to read the message. So we have our large and small ribosomal subunit made out of rRNA. The cool thing about tRNA is it kind of looks like the letter T anyway. So what you're going to see is we're going to learn about codons and anticodons when we come back. But the T is kind of like, this is all tRNA. And what you'll find out is there's a region that we'll learn about called the anticodon. And on the other side is where our amino acid is going to attach. This tRNA is only specific for one amino acid. That's it. Have you guys noticed that we have a lot of things that are very specific in our body? It doesn't work for any, one enzyme only works for one reaction, nothing else. Same thing with this. One tRNA only codes for one amino acid. So you have to have how many tRNAs floating around? 20 different amino acids, we said, right? So you have at least 20, you have many of them, like many copies of the one tRNA. But you have to have 20 different tRNAs to bring the right amino acid that your message is telling you it needs to build your proper polymer, right? Your protein. So we'll go into those details of the translation going from the RNA, the nucleotide message, into a amino acid language, right? So we'll come back to that later. If you guys want to practice what you think before we come back, I know you're focused on exam 1, but if you want to practice, I wrote what kind of strand here?
DNA.
I wrote a DNA strand there. You can take a look at that strand and create a messenger RNA out of it. Don't worry about splicing and capping and tailing it, but you could write the word cap and tail on either side, okay? And then we're going to go through that process when we come back and make areas called a codon, and we're going to talk about where the anticodon is, and we'll finish up that story of gene expression. And then we'll get into cell growth and reproduction and tissues and things like that, so we can tie up the rest of this